Good day everyone. Before this, we all have learned about glycolysis and the Krebs cycle. So today, come with me and let's dive into the last process, which is ETC. Don't forget to enjoy the video.
ETC or electron transport chain is the biochemical process that produces most of a cell food in an aerobic organism. This involves the build-up of proton motive force or PMF, which allows the production of ATP, that is the main catalyst in cellular reaction. But I wonder, who is the first person to discover this electron transport chain?
Me. In 1948, Albert Lanier discovered with Eugene P. Kennedy that mitochondria are the site of oxidative phosphorylation in eukaryotes, which ushered in the modern study of energy transduction. In 1961, he discovered that the citric acid cycle and the electron transfer chain of enzymes where one NADH makes 3 ATPs are located within each cell's mitochondria. Most living cells produce energy from nutrients through cellular respiration that involves the taking up of oxygen to release energy. The electron transport chain ETC is the third and final stage of this process, the other two being glycolysis and the citric acid cycle.
There are two agents, reducing and oxidizing. The chemicals that are oxidized are reducing agents. They donate electrons and reduce the other substances by taking their electrons. While oxidizing agents, they accept electrons and oxidize the other parties in the redox chemical reaction.
Cellular respiration involves many reactions in which electrons are passed from one molecule to another. Reactions involving electron transfers are known as redox reaction, and they play a central role in the metabolism of a cell. When there are a series of redox chemical reactions taking place, electrons can be passed on through multiple stages until they end up combined with the final reducing agent.
Eukaryotic cells have a nucleus.
Meanwhile, prokaryotes doesn't have mitochondria.
The electron transport chain reaction takes place on and across the inner membrane of the mitochondria.
ETC reactions in prokaryotes take place in the cytoplasmic membrane.
ETC uses high-energy electrons from chemicals produced by the citric acid cycle and takes them through 4 steps to a low energy level. The energy from these chemical reactions is used to pump protons across a membrane. These protons then diffuse back through the membrane.
For prokaryotic cells, proteins are pumped across the cell membranes surrounding the cell. For eukaryotic cells with mitochondria, the protons are pumped across the inner mitochondrial membrane from the matrix into the intermembrane space.
Chemical electron donors include NADH and FADH, while the final electron acceptor is oxygen. The chemicals NAD and FAD are given back to the citric acid cycle, while the oxygen combines with hydrogen to form water.
The protons pumped across the membranes create a proton gradient. The gradient produces a proton motive force that allows the protons to move back through the membranes. This proton movement activates ATP synthesis and creates ATP molecules from ADF. The overall chemical process is called oxidative phosphorylation.
Before we dive into these complexes functions, let us know more about each of these complexes. 4 chemical complexes make up the electron transport chain, and each of them has a specific role of its own.
To start, 2 electrons are carried to the first complex about an ADH. Complex 1 takes electron donor and ADH from the matrix into the intermembrane space and sends electrons down the chain while using the energy to pump protons across the membranes. Complex 1 can pump 4 hydrogen ions across the membrane. It is in this way that the hydrogen ion gradient is established and maintained between the 2 compartments separated by the inner mitochondrial membrane. Then, there is the second complex. Complex 2 uses FADH2 as an electron donor to supply additional electrons to the chain. Note that it does not pass to complex 1. Molecule Q. It receives the electrons derived from the NADH from complex 1 and the electrons derived from FADH from complex 2. For what? To deliver the electrons to the next complex in the ETC.
Next, we have complex 3 that passes the electrons to an intermediate chemical called cytochrome C proteins, or also called cytochrome oxidoreductase, and pumps more protons across the membranes to the fourth complex. Complex 4 receives the electrons from the cytochrome and passes them on to half of an oxygen molecule that combines with two hydrogen atoms and forms a water molecule.
At the end of this process, the proton gradient is produced by each complex pumping protons across the membranes. The resulting proton motive force draws the protons through the membranes via the ATP synthase molecules.
As they cross into the mitochondrial matrix or the prokaryotic cell's interior, the action of the protons allow the ATP synthase molecule to add a phosphate group to an ADL, or adenosine diphosphate molecule. ADP becomes ATP, or adenosine triphosphate, and energy is stored in the extra phosphate bond.
Cut! Alright, take 5 everyone.
Together with me today is Shake and Kausa. They will be explaining to us the importance of ETC and what will happen if ETC does not occur. So, Shake, can you explain to us the importance of ETC?
For sure, each of three cellular respiration phases incorporates important cell processes, but the ETC produced by far the most ATP. Since energy production is one of the key functions of cell respiration, ATP is the most important phase from that point of view. The other key function of the ETC is to produce NAD and FAD from NADH and FADH in the first two chemical complexes. The product of the reaction in ETC complex first and complex second are the NAD and FAD molecules that are required in the citric acid cycle. As a result, the citric acid cycle is dependent on the ETC.
Okay, wow, what a great explanation. Thank you, Shake. Now, moving on to Kausa. So, what happens if ETC does not occur?
Alright, thanks Adrianna. So, what happens if ETC does not occur? So, if you prevent electron transport, you prevent proton translocation. The event must happen together or not at all. So, if oxygen isn't there to accept electrons, for instance, because a person is not breathing in enough oxygen, the electron transport chain will stop running and ATP will no longer be produced by chemiosmosis. ATP cells can carry out the reactions they need to function, and after a long enough period of time, they may even die.
I see. Thank you, Kausa. So, alright, viewers, that's all. Wait. Oh, before that, I forgot. The video isn't finished yet.
To conclude what we have learned in this video, the electron transport chain is the portion of aerobic respiration that uses free oxygen as the final electron acceptor of the electrons removed.
Not only oxygen, but also sunlight and other requirements. In Surah At-Takwir, verse 18, A'udzubillahi minasyaitonirrajim, As-subhi idza tanaffas, which means and the dawn as it breathes. The term dawn in the verse refers to the time when sun rays begin to reach the earth. The plants will undergo the process of photosynthesis only if they receive those rays. But why does it only state dawn instead of daylight itself? Well, as we know, this phenomenon of photosynthesis occurs only during the day,
or to be exact, when there is sunlight present. Since it is dependent on the energy of light and cannot be realized in the dark. So, when the dawn breaks, as described in the verse, means that at the beginning of sunlight illuminates the earth and signals the plant to start the photosynthesis. The light shines and oxygen liberated. Don't we think, how can a book that was revealed centuries ago be able to express this such extraordinary scientific evidence of the process? This makes clear the reason for successive usage of word dawn and breath in the verse. Therefore, we can see that there are various requirements for plants to undergo the process of photosynthesis, thus giving us oxygen to breathe.
Well, viewers, we have reached the end of our video. That's all for today. Join us again in next classes where we learn more topics in biology together. Finally, I am Adrianna Alisa Muhammad Aris and all team members of ETC Group. We say, see you next time. Wassalamualaikum warahmatullahi wabarakatuh.
Meanwhile,
Alongside the evolution of humankind, many planting industries are evolving as well. In assuring the level of crop cells, they use plant pesticides to get rid of parasites. Most plant pesticides are chemical-based and have affected the crops as a whole. So, if it causes a threat to the products, why don't we incorporate nanotechnology in the production of the crops? We can apply nanotechnology in agriculture. How, you might ask? They can act as sensors to monitor the consistency and condition of the soil in agriculture fields. It can also monitor products and nutrient levels to improve productivity while preventing soil and water degradation. It also provides protection against a variety of insect pests and microbial disease. As a result, it maintains the health of agricultural plants.
In Suratul An-Nahl, verse 13, Allah has stated, وَمَا ذَرَأَ لَكُمْ فِي الْأَرْضِ مُخْتَلِفًا أَلْوَانُهُ إِنَّ فِي ذَلِكَ لَآيَةً لِقَوْمٍ يَذَّكَّرُونَ. Sadaqallahul Azim. Means, and there are also signs for those who take heed in the numerous things of various colors that He has created for you on earth.
I see. Thank you, Kausa. So, alright viewers, that's all. Hello? I'm very sorry, but the video isn't finished yet. Assalamualaikum warahmatullahi wabarakatuh and good morning to everyone.
Is the portion of aerobic respiration that uses free oxygen
as the final electron acceptors of the electrons.
Sini je nak nampak electron.
Tak ada nampak. But I wonder who is the first person to discover this electron transport chain?